Today we are going to learn about the structure of a typical eukaryotic cell. Before moving to the topic, we should know who has first observed it or found it. Robert Hooke in 1665 observed dead cells in the cork under compound microscope. With the discovery of electron microscope, Anton von Leeuwenhoek first saw and described live cell in 1676. In the previous video, I have already discussed about the definition, structural unit, functional unit, discovery of cell, as well as cell theory. If you want to watch it, you can. Today we are going to learn about the structure of typical animal cell, right? So outermost covering is the plasma membrane. It contains finger-like projections that is known as microvilli. Inside the plasma membrane there is cytoplasm, and cytoplasm contains all these organelles inside it, right? So we are going to see one by one about all these organelles. First of all, plasma membrane or cell membrane. So cell membrane is the boundary. You can say in simple language, it is a boundary. It separates a cell from the outer environment. So it is thin, transparent, living membrane, semi-permeable. So few materials can go inside and few materials can go outside according to its requirement. So cell membrane protects cell. It gives definite shape to the cell, and it is composed of lipids and proteins. Next, it is the nucleus. So what is nucleus? Nucleus is present inside the cell. It is the controlling unit. You can see it like this is the cell, right? It is the outermost covering, that is plasma membrane, and this one is the nucleus, and this is membrane bound, and its membrane is called nuclear membrane. Its membrane is called nuclear membrane. Inside it you might be saying this is the small granular part. It is the nucleolus, and as the cell has cytoplasm, nucleus has nucleoplasm, right? So these are the structure. Here in this picture you can see this is the nuclear membrane or nuclear envelope, right? And in the nuclear membrane, these are the pores. Whatever you are seeing, this small black one, these are the pores. It helps in transporting the material from inside to outside. Okay. So nucleus is the most important part of the cell because it is the control unit, and it contains the genetic material. Genetic material is composed of DNA and proteins. It involves in protein synthesis, growth of cells, cell division, and differentiation, right? In the nucleus you have seen the nucleolus. Nucleolus is not bound with any kind of membrane. It is composed of RNA and proteins. It rewrites rRNA, that is ribosomal RNA, and combines it with the proteins to form incomplete structure of ribosomes. This is then transported out to the cytoplasm where complete ribosomes are synthesized. So nucleolus is involved in the synthesis of part of the ribosome, and ribosome is responsible for the translation of RNA into the proteins. Microvilli. These are the finger-like projections from the plasma membrane. So what is its function? It increases the cell surface area. And you can observe it in the epithelial cells of the organ or the organ systems, where it performs the function of absorption of molecules. Example, in the lining of small intestine. Small intestine need to absorb materials so that it can do the digestion process properly. So in the lining of a small intestine there is epithelial cells such as columnar epithelial cells, which has microvilli, which increases its surface area but not its volume, or you can say its size. So it is very useful in this case. Now mitochondria. Mitochondria are the double membranous structure present inside the cell. It is very important organelle as it synthesizes ATP. ATP generates energy, right? So it contains all the enzymes which are necessary for the oxidization of food. Food gets oxidized and finally it produces ATP. You can see its structure. This is the inner membrane that is folded, and because of its folding it forms cristae and matrix, right? This is in foldings, it is the cristae and matrix, and outer membrane is the simple membrane. Peroxisome. What is peroxisome? It is the another organelle of the cell. Its outer membrane is just like the plasma membrane, and inside it is the crystalline core. Crystalline core contains oxidative enzymes, mainly urate oxidase, catalase, D-amino acid oxidase. Hydrogen. It helps in hydrogen peroxide metabolism, because hydrogen peroxide, that is H2O2, is synthesized and it is dangerous. Again it is break down into hydrogen and water molecules. So peroxidase helps in the hydrogen peroxide metabolism, as well as fatty acid oxidation, degradation of purines, polyamines and amino acids. Moving to the endoplasmic reticulum. So endoplasmic reticulum is the continuation from the nuclear envelope to the cell membrane, and it forms the network like structures, right? So it divides the cytoplasm into inner luminal and extraluminal cytoplasm, and it helps in the transportation of materials throughout the cell, which is very essential. So you can see here these are of two types, that is the rough endoplasmic reticulum and smooth endoplasmic reticulum. So these are the structure. Immediately after the nucleus, this is the nucleus, and in continuation it has rough endoplasmic reticulum. So what will happen? DNA is synthesizing what? mRNA, and mRNA should be translated, and where it gets translated? On the ribosomes. Rough endoplasmic reticulum contains ribosomes, small granular, you might be seeing here, small granules. So these are the ribosomes. These ribosomes are very important for the synthesis of protein. After that, you can see smooth endoplasmic reticulum. Smooth endoplasmic reticulum does not contain any kind of ribosome. It helps in the transportation of materials throughout the cytoplasm, and it is the site of lipid synthesis. Now, Golgi apparatus. It is the another organelle of the cell. It is these stacks of thread-like thin membranes of variable shapes and sizes. Right, you can see here it's of different shapes and sizes. It involves in packaging of proteins and lipids. As you know, rough endoplasmic reticulum forms proteins and smooth endoplasmic reticulum forms lipids. These proteins and lipids in the form of vesicles get enters into Golgi apparatus. Golgi apparatus then sorted these proteins and lipids and package it, form it in the final structure. Right, so proteins and lipids get final structure in the Golgi apparatus, and it is delivered. Proteins and lipids will be delivered to the intracellular organelles, other intracellular organelles, wherever it is required. It is secreted outside the cell. It is also secreted outside the cell. It is the important sites for the formation of glycolipids and glycoproteins too. Now, lysosomes. Lysosomes, you might have heard lysosome is called the suicidal bag. Why is it called so? Because it contains hydrolytic enzymes. Hydrolytic enzymes break down carbohydrate, lipids, proteins, nucleic acids, and these enzymes are lipases, proteases, carbohydrases, and these enzymes are active at acidic pH. Whenever something abnormal happens inside the cell, or it is infected with bacteria or viruses, these hydrolytic enzymes acts upon these viruses, bacteria, or the abnormal cells. It may act upon itself, means the cell in which it is present, and finally the whole cell will die. So it is like committing suicide. That's why it is called suicidal bag. Right? So this is all about the structure of animal cells. In the next video, we will learn about the structure of plant cell, and we may compare it with the animal cells. Thank you.